You hear me? Josephine, where's the dongle? Thank you very much, everyone, for being here to my PhD presentation. But before starting, I need to mention that, especially on the paper side of the thesis, there are some mistakes that I'm going to fix them with an errata and publish it and give it to the committee members and the opponent and everyone who needs it online. As you can see, thank you very much for the introduction, Kevin. My presentation, my thesis title is Dynamics and Interactions in Entangled Nanofiber Dispersion. As you can see here, there are beautiful examples of fibrous networks in our surrounding environment. We can find them everywhere almost. In biological systems, like in our body, in the bones, in the skins, in the cells, and even in the plant cell walls, we can find these kind of structures. But due to the properties that they have, scientists try to replicate them in the labs and make the best use of them. If we see here, if we see this picture, this is a picture of a cell which is trapped in the collagen network. And as we can see here, we can see that the cell is interacting with the collagen network, and also it tries to diffuse in the system. So these kind of structures can bring structural integrity for the environment and also open a network for the diffusion in the system. And a drug, for example, or nanoparticle or any kind of molecules can diffuse into these systems. But one thing that we need to remember here is that these systems are dynamic. They have thermal motion, and due to inherent flexibility of the fibers, they can have different behavior or different rheological behavior with the surrounding environment. And when they are crowded, they can show collective behavior that they can be different compared to a single semi-flexible fiber. So the first thing that we need to know is about the thermal motion or Brownian motion of the fibers. Albert Einstein established a mathematics behind it, but he established it for a spherical particle. And when we have a spherical particle, it easily diffuses into the system. But in our cases that we have a particle with high aspect ratio, and it's not just translational motion, it's translation plus rotational motion, which these two are highly coupled together. They affect each other. And even we can add more complexity into the system when we go with higher concentrations. And as I mentioned before, the fibers have inherent flexibility. So these parameters can affect the dynamic more into detail. So here we have Professor Dejean explaining reptation theory. And reptation theory is, if I want to explain it in a simple way, is that if we have a bowl of spaghetti and we try to take out one of the spaghetti, we can easily do it. But if we try to grab a bunch of them with a fork, we need to imply high amount of force. So, but this reptation theory, who was established by Professor Dejean, was developed for flexible polymers. And the other assumption was made that there was no interaction between the polymers. And we had these assumptions that only the labeled polymer was moving in the system. So the surrounding were considered a static system. So during the years, scientists tried to improve the model to be applied for different kind of polymers with different flexibilities. So here on the right side, we have Doi and Edwards, who were considering that flexibility doesn't affect the rotational motion of the fibers. On the left side, Professor Dejean, that he predicted in his book that flexibility can affect it, and Professor Theodik, he proposed the theory behind it. So in 2010, Michael Rubinstein published a review paper, and he asked the scientists in the community to come up with some solutions with the questions that they are still there. For example, there was no any definition, like a specific definition for the entanglement or the tube diameter that we had in the reptation theory. Or even there was no any particular way to reach from dynamic of the system to the rheological behavior. So what is important here is that flexibility matters. So it's consider a polymer in water, and the thermal motion try to bend the polymer, but on the other side, the inherent flexibility try to push back the polymer to its shape. And just imagine here, these polymers which are rigid, semi-flexible, and flexible one in a really confined environment. So definitely they will have different behavior. So if I compare my thesis with a house that I wanted to build, paper 1 and 2 were on the stage that I wanted to buy the land. And with the help of my supervisor, I was introduced to the project, and I kind of find my way to be able to do what in the research in the field that I wanted to do. And paper 3 is like a foundation for my system, and paper 4 and 5 is like the exterior design of the house or the thesis. So here, since I'm the main author in the paper 3, 4, and 5, I will focus on these 3 papers. So the first one is the effect of stiffness on the dynamic of entangled nanofibers. And here we have done a wide range of simulations with different parameters to come up with a solution or improve the model for the dynamic reptation theory. So this is the details related to my simulations. So I have used hardcore potential, Weeks-Chandler, Anderson potential, and I have built the polymers with bead spring models. So we define the beads and we connect the beads with the spring together. So I have used harmonic interactions for the springs to put the beads together, and angle potential to imply the rigidity or flexibility of the fibers. And one thing that we have here is that we have coupled the molecular dynamic or coarse-grained simulations to the lattice-Boltzmann fluid to take into account hydrodynamic forces that we have over there. And for this, I have used Espresso MD package, which is a package established in Max Planck Institute, and now it's in Stuttgart University. So there's some more details on the simulations. To capture the reptation motion, we need to go for crowded environments. So almost all of the cases that I have are crowded. And I have a cubic simulation box, periodic in the 3 directions. And the box length is a bit larger than the fiber length to be able to get the right dynamic, especially for rotational dynamic of the fibers. And the procedure is this, that we introduce the beads into the box. We set the springs according to the persistent lengths that I define for the polymers, and we warm up the system. And then when the polymers get the shape, we couple them with the hydrodynamic forces, and we thermalize the whole system together and prepare it for the production. But there are some challenges that it's a time-consuming job, because when you have a flexible polymer, you need more time for the warming up the system to let it get the shape that you want. And of course, concentration also affects it for warming up and also the production one. And when we have crowded system, random interaction of the beads also can be difficult. And coupling the beads to the fluid that I wanted, it's a non-trivial job, and you need to try different parameters for it. So the parameters that we have, of course, is the spec ratio, the concentration, which I have defined it compared to the mesh size of the, relative to the mesh size of the system, and of course, persistent lengths. And for this one, we have used, there are so many articles in the literature, but we have used this one that they have established a system that they look at the cellulose from polymer physics point of view, and they have measured the persistent lengths considering the electrostatic interactions. And the observable that I have chosen, since I want to analyze the rotational dynamic, I calculate the mean square angular displacement and orthogonal mean square displacement, which is the normal deflection of the fiber that we can get tube diameter out of it. And for analyzing each fiber individually, we define end-to-end fluctuation of end-to-end vector to see when the dynamics of the fibers relaxes. So for the validation of the cases, since I have wide range of cases, different concentration, different flexibility, I have used different resources. For the rigid rod particles, I have used Doi and Edwards' book and all the papers related to this topic from them. And when it comes to effective flexibility, since Professor Odik has established this theory, I have used this paper. And as I mentioned, when we have a slender particle, it diffuses differently in parallel direction or normal direction. So here they have a really good theory that I have used that. So the first thing that we need to know, if we go back to the paper, they were interested in quantifying the polymer entanglement and the tube diameter. So here we use orthogonal motion of the fibers to see how it deflects in normal direction. So if we consider the yellow case is in this figure, the yellow case is a low concentration. As you can see, the normal diffusion is linearly growing. But when we step by step increase the concentration, we can see that there is a behavior towards plateau behavior, that it tells you that the fibers are entangled or in an imaginary tube, and all of a sudden we have a super diffusive motion that it shows that the fiber releases itself and diffuses into the environment. And with the help of this, we can also quantify the onset of entanglement. So between linear and plateau, we find the point or the case that it starts beginning of tubing effect. And if we do this for many cases, we come up with the inset image here. That the image tells us that, okay, if we have two samples of polymer or cellulose nanofiber with the same concentration but different flexibility of the fibers, the one with the higher flexibility can provide entanglement more compared to the one with less flexible one. The left side, you see the ones with higher flexibility, that it can be visible that it has more entanglement. So the next parameter that which is really important for us is the mean square angular displacement, and it's related to largest relaxation time of the system or terminal relaxation time. And this is important because with the help of this, we can get the rotational diffusion, the parameter that we can compare with the literature. And it's inversely proportional to rotational diffusion. So in 2010, a paper published from Fakhri Group in MIT that they prepared an agarose gel, and they released a carbon nanotube in it with different flexibility, and they realized that Professor Odik was right. If you have a more flexible polymers, it increases the rotational diffusion of the polymer. And they compared with the Doi theory and Odik theory, and they came up with this, that Odik theory was more efficient or more precise in considering the effect of flexibility. But again, they had a static gel, agarose gel, and they didn't talk about the onset of entanglement. But if we have a case that all the polymers are moving and all of the polymers are showing the thermal motion, this is what exactly happens. This is a cross section of the center of the box that I have. And you can see that these are empty spaces between the polymers. And you can see the tubes, they appear, disappear, and they are not stable at all. And when we consider this and plot it and get the rotational diffusion, we can see on the figure on the right side that the yellow one is the one that we have proposed. Actually, for the rigid ones, Doi, for the rigid particles, rigid polymers, Doi theory works, but for the semi-flexible one, we have proposed neo-scaling for rotational diffusion. So the other question that I was asked is that how we can relate it to the rheological behavior of the system. And the thing is that we, in most of the simulations that I had an extra bead into the system that was diffusing into the system to get the trajectories and use it for microrheology purposes. And here we did the microrheology, and we got G prime, G double prime, storage and loss modulus, and then got the viscosity of the polymers. And here there must be a citation related to the experimental results, which is from Malin Nordstrom. And we compared the viscosity that we got with, we compared with some experimental results that was published in the community, in the WWSC community that we had. So here I move to the second paper, or paper 4, which we consider the effect of charges into the system. And this paper is under revision in ACS Nano, and we got the first round of revision. So at this point, we thought about it that, okay, since we have the system, maybe it's better to make it more complex and make the best use of it. So we added charge, and if we look at if we look into the literature, there are so many questions that they are still unanswered related to fibrous networks and with different applications. But one of the things that I found is that charge is really important in drug delivery system in the fibrous networks. And there is a fact that only 0.7% of the drugs or the nanoparticles can reach the target. And the key players here are steric hindrance, electrostatic interaction, and hydrodynamic forces, which are related to parameter like size, particle size, or concentration, and the system charge, I mean, the charge of the fibers, and also charge of the nanoparticles that we have. So what we did here is we had a system, we had hindrance, we had steric hindrance, we had hydrodynamic forces, so we added electrostatic interaction into the system with the help of particle mesh, or P3M algorithm, which is in Espresso. And the parameters that we have is network concentration, relative surface charge, density of nanoparticle to the polymers that we have, and also the nanoparticle size. And we have some observable here to like diffusion coefficient and permeation lengths and the time that it gets for the particle to reach the target, or mean first passage time of the particles. So the first thing that we did is to get the diffusion coefficient of the particles with the help of Kubel-Green function that we have. With the velocity autocorrelation function, we can get the diffusion coefficient and compare it with the other cases. But honestly, from diffusion coefficient, we cannot get anything, or it's useful, but not really decisive. So we plotted the contour plot for relative diffusion of the particle compared to the case that the particle is diffusing into the system without any obstacle, or in really dilute systems. And what is interesting is that we saw a universal behavior in them. And the combined effect of charge and concentration on D over D0, or relative diffusion, is independent of the size. So what we did here is to improve the study with some experiments, which I have done in Switzerland at EMPA. We were interested in to see how far the nanoparticles can permeate in the charge network. So we use cellulose nanofibers, which are negatively charged as a system, and we released some charged nanoparticle into the system, positively and negatively, to see how they diffuse into the system. And as you can see here, the point that we introduced the nanoparticle, since they have different charges, at the beginning, there is a complexation layer, and when it grows, we give it some time to let the nanoparticle diffuse into the system, and we measure the size. So here, we compare exactly these experiments with the simulations, and all of a sudden, we released the nanoparticle inside the network. And as you can see here, we have neutral nanoparticles and positive nanoparticle in negatively charged network. And as you can see here, that with considering the effect of concentration, is that the positive nanoparticles can diffuse with lower rate. So we measured this size, permeation lengths for different cases, and we plotted for the simulations and experiments that we have, and we got almost not the same results, but at least the trend is the same. But honestly, even the permeation length is important, but not as important as this one. One of the most important parameters in drug delivery system is the time. So maybe we insert a drug and it takes 2 days or 1 minute to reach the target. So we have the network, we define a target, and we release the nanoparticles, and we see how long it takes for the nanoparticles to reach to the target. So we plotted the mean first passage time for different cases, but honestly, if you plot it like a heat map, you can see that charge has a effect, but we can see that it's not really linear what is happening. So what we did was to do some random forest model analysis to see what is the contribution of each parameter, size, charge, or concentration. So what we found here is on this right box plot that we can see the importance of the features that we have. And as you can see here, charge is almost as important as concentration, or even more than that. But what is the fate of nanoparticles? So what if we release a nanoparticle, what can happen? So we have done some image analysis and also video analysis that when we release them, there are three main regimes that they can happen. They can stick to the fibers, they can slide on the fibers, or they can be bouncing inside the fibers. So we defined these regimes based on the normalized attachment time of the nanoparticles to the fibers. And each of these can be beneficial for different applications. For example, when you have a sticking behavior, it's beneficial for filtering, and sliding, it can be beneficial for cargo delivery inside the cells. And bouncing is to reach a cell further than the extracellular matrix. So here we plotted the NAT, or normalized attachment time, and we defined the regimes that they are there. And we found that the ratio of the nanoparticle size to the mesh size is important. And when this amount is around 1.2, we can see that there is a difference in dynamics over there. And at that point, electrostatic forces can be beating or overcoming the hydrodynamic forces. So the last paper, which is an ongoing work actually, is the modeling of hybrid networks. When we have different polyelectrolytes and we mix them, we get hybrid networks. And in this case, I focus on cellulose nanocrystals and amyloids. So what happens when we have oppositely charged polyelectrolytes? We mix them. And of course, since we have different charges, we have attraction. And there is a phase separation here that the dense one is more condensed, that we call it coacervate phase. And the other one, which is a mix of the ions or isolated polymers, they are the dilute case. And this simple mechanism is so useful in the industry. They can naturally happen in our body and in food industry, drug delivery system, even vaccine formulations, and cosmetics, water treatment. So actually last year I visited EMPA in the lab of Professor Gustav Nystrom, and they had a published paper that they mixed amyloids, whole proteins of the egg with CNCs and CNFs. And they did some structural analysis and rheological analysis to see what happens when we mix these two together. If we see in the literature, there are many excellent experimental work on this subject, but there are limited models. I mean, there are many models, but limited ones that they are famous. And I have used this one, which is published by Michael Rubinstein. So here, if I want to compare it with the previous simulations, the simulations are almost the same, except the length scale that I have. So I have different parameters like charge fraction, Kuhn length, number of monomers per blob, and concentration. And with that, we can define electrostatic blob size. And electrostatic blob size is if we consider a segment of polyelectrolyte and it interacted with itself, I mean, exactly the same segment of another polyelectrolyte, when the electrostatic energy is comparable with the thermal energy that we have in the system, this is the blob size. And the thing is that when you have a stronger polyelectrolyte, you have a smaller blob size because you have more concentrated charges on the polymer. And in the paper, they they propose correlation lengths of the system, which is the mean distance between the polyelectrolytes when the counter ions are between them. So what I did was to create a simple system with polyelectrolytes and its counter ions and let it go and measure the correlation lengths of the system. And we define the correlation lengths of the system with defining some points on the polymers and make them bigger step by step until reaching to the next imaginary polymer. And I plotted with the, to compare with the theory that there is proposed by Michael Rubinstein. The lines are from theory and the rest is from the simulations. And exactly like the experiment that they did, they had like two different kinds of polyelectrolyte, hen egg white lysosomes, HULE, with positive charge. This is zeta potential. And cellulose nanocrystal with the negatively charged. And we did some calculations and we came up with these sizes for the blob sizes that we had over there. And of course, the amyloids had some flexibility and size distribution. And for finding those distributions and details for designing the amyloids, I have used this paper. And interestingly, the thing is that even flexibility, except the sizes a little bit, the width size, everything else looks like or close to cellulose nanofibers. So what we did here is to define a system with cellulose nanocrystals with and amyloids to interact with each other and see how they change, how they create new structures. So this is the image. The image that we see here is exactly the sample from EMPA that we can see that there were two liquids, and now we have a material with different rheological behavior. And we do some structural analysis. When we find the structures, the bundle structures that they create, and we've compared with the images, we can see some similarity between them. Like the cluster, they can create clusters of amyloids and CNC together, and even the fibers can in a parallel way, can be attached together if we have CNC over there, or even we have cross-shaped assembly over there. And except the structural analysis, we did some rheological analysis. Again, I added a nanoparticle into the system comparable to the mesh size of the system, and to see how it diffuses and get the trajectory and do the macrorheology. And we got the G prime, and we estimated the G0 amount and compare it with some published results that you can find it in literature. And the slope of the line here is a little bit different. The reference one is 2.3, but 1.99 or 2 is the one that I have got at this point. I try to improve it, but this is the stage of this project. And actually, this is the beginning of this project, and I would say it's kind of validation for this case, for this system. And this work, of course, is going on. So if I want to conclude it, I would say what we did in the third paper was creating a basic for the system that we had, and we found some interesting results related to rotational diffusion and tube size diameter in the semi-flexible cases. And we were able to compare it with the results that are published already in the literature. And then in the next work, the